I think it's time. We'll go ahead and get started. It's my distinct pleasure to introduce to you all today Robert Huff, or should I say Robbie, as maybe he's known to his family and close friends. Robert is a fourth-year graduate student in biology. He's the first from my laboratory to defend their PhD thesis. So we're proud of him for that. He's originally from California. Robert holds a Bachelor of Science and a Master of Science degree from San Diego State University. While he was there, he studied virology in the laboratory of the late Dr. Roland Volkovich. I would say to say that Robert has been productive at Baylor would be a real understatement. He's the first author of 2 peer-reviewed publications. He's a co-author of an additional 2, and we are currently drafting 3 more manuscripts that he'll be the first or co-first author of. So that's an incredible record in my laboratory, and certainly that's really set us up for success long term. So he served as a teaching assistant and also as an assistant laboratory coordinator in biology. He's presented his work at 5 professional conferences, and this semester he is the recipient of a certificate of excellence in research from the Department of Biology. In 2019, he was also given an award or recognized by the Texas Mosquito Control Association for an outstanding graduate student presentation. On a personal level, I would just like to say Robert he's tremendously talented. I promised him I'd try not to embarrass him today. He's naturally inquisitive. He's absolutely welcoming and helpful to everyone in the lab. He's been a real leader for our lab. He's dedicated to self-improvement. And he is perhaps one of the best problem solvers that I've known. And that's a really important quality, as all of you know, when you're in experimental biology. Truly, he's been a major contributor to all of our collective successes. So Robert, for those efforts and for your friendship, we really are truly grateful. Assuming things go well today, Robert is going to embark on a new academic journey. He has accepted a position at UC San Diego in the laboratory of Dr. Eniko Sajti, and he will investigate the molecular genetics of a lung disease called bronchopulmonary dysplasia. So that will be, I think, really exciting, and I'm sure that he's going to make great contributions there. For today, the title of Robert's dissertation is, as you can see, Functional Characterization of Olfactory Receptors and Molecular Species Identification of Vector Mosquitoes. Robert, the floor is yours.
Thank you. Thank you all for coming in person and virtual. Hopefully this clicker works now. Yes, okay. I'm assuming we'll stand here and we'll be on this screen. So I just want to start off by stating the obvious. Mosquitoes are deadly vectors of disease. This is a little figure from the NIH. So as everyone knows, they can transmit a number of different viruses, protozoas, and different nematodes, typically in the form of filarial worms. So that I just wanted to put out there. That's why we do the research we do, is because they cause widespread morbidity and mortality throughout the world. Mosquitoes are also extremely invasive. In particular, their zones of habitation are going to increase dramatically, as predicted by this paper in Lost and Neglected Tropical Diseases. So what this group did was modeled where this mosquito Aedes aegypti is found throughout the world, and they're trying to model what they think is going to happen in response to climate change over the next 60 years. So they predict, using their model, that there's going to be a dramatic increase in the northern latitudes of where this specific species is going to be found. As you might expect, the greater the range of mosquitoes throughout the world, you can hypothesize that there's going to be an increase in disease transmission in these zones as well, due to the expansion of their zones in which they have. Mosquitoes are also durable, so they have become more and more insecticide resistant throughout the years due to evolutionary changes that occurred in response to insecticide usage. So that's one of the primary mechanisms of reducing mosquito populations throughout the world. And this occurs through a number of different mechanisms that I won't go into detail, but this is a really big problem throughout the world, not only for mosquitoes, but also insects in general. As insecticides become less and less effective, we're going to see more and more population growth of different types of insects. So I just want to kind of give you an idea of how this talk is going to go. So I'm going to start out with some brief background on chemosensory biology, and then I'll go into floral odor detection by mosquitoes and a different insect, as well as host odor detections, and then I'll finish it off with some species identification of insects. So without further ado, I'll go ahead and get started. So our lab focuses on the chemosensory system of different insects and their associated behaviors that are caused by some molecular interactions between receptors and ligands found in the environment. So one thing we focus on is nectar feeding. So both male and female mosquitoes require nectar sources in order to obtain carbohydrates. Female mosquitoes oviposit or lay their eggs, and the larval development and of the immature stages of mosquitoes is also dependent on chemosensory mediated behaviors. Mosquitoes also mate and utilize this system, as well as find blood meal hosts. So all of these behaviors are important to the fitness of the vector, and it's things that we study in the lab. So I'll kind of generally explain how the chemosensory system works in these insects. So both adults and immature stages of mosquitoes have a number of different sensory appendages. Here we're showing you an antenna from the adult mosquito that is decorated with those small hair-like structures known as sensilla. Inside of the sensilla reside olfactory receptor neurons, and on the membrane of these receptor neurons are a number of different receptor proteins that act as ligand-gated ion channels. So what happens is an odorant that is inside of the air or outside in the environment can pass through into the sensilla, where it can bind to a receptor. When this occurs, this protein acts as a ligand-gated ion channel. Ions flow into the inside or the cytosol of the neuron, leading to an action potential that travels to the insect brain, and ultimately some sort of behavioral output comes of this. So we typically focus on three main classes of receptors. So we have ORs, or odorant receptors, in mosquitoes and other insects that require a obligate co-receptor, orco, in order to function as a receptor complex. We also study IRs, or ionotropic receptors, that generally require at least one co-receptor. These are a little bit more complicated, and I'll talk a bit more about that later. And GRs, or gustatory receptors. So these are typically associated with tasting or contact chemosensation, and I will not be talking about these today, but I just wanted to let you know that there's also this additional group that is very important to the vector itself. So to study these specialized proteins, we use a protein expression system using Xenopus laevis oocytes. I think I have to click on this before I can use the clicker. In order to study different classes of receptors, so ORs, IRs, GRs. So what we do is we microinject these frog oocytes with messenger RNA transcripts that code for our genes of interest. So they're injected into the cytoplasm of the oocyte, where they're translated into functional membrane proteins that travel to the cell surface of the oocyte. And what's unique about this is they function the same way they do in an adult mosquito's neuron. So they act as ligand-gated ion channels, or odor-gated ion channels is another way to think of it. So the way we can record from these functional membrane complexes is using 2-electrode voltage clamping, which is an electrophysiology technique where you use 2 micro electrodes that pierce the cell membrane of an oocyte that is expressing our proteins of interest. So basically what happens is these receptor complexes are typically in a closed state, but when an odor binds to the receptor, the channel will open, allowing ions to travel inside of the oocyte, and this leads to a measurable current using this technique. So we express these receptor complexes, and we can perfuse different odors across them and see whether or not they open in regards to the changes in current. So we have a chemical library that was developed in the lab, not developed, but curated in the lab, of about 200 different compounds. And these compounds are known in the literature to impose some sort of behavioral change on insects in general. Maybe not necessarily mosquitoes, all of them, but insects in general. So we dissolve these odors in a perfusion buffer and pass them over the surface of the oocyte. And if we get a response to one, it can look a little bit something like this. So at the top, we have a baseline current. And once this oocyte gets stimulated with 1-hexanol, we get a dramatic drop in current that will basically recover to baseline, and we could stimulate again with another compound. So from this digital information, we're able to basically convert it into something like this that's easy to read. So this is an example of doing a blend screen. So this is typically how we start a deorphanization experiment. We group all of our chemicals from our library into groups of about 10 or 12 based on chemical structure. So that way we don't have to do all 200 compounds one after another, because that would take much too long. So for example, alcohol blend number 2 is giving a really good hit here. So the next step would be to take this alcohol blend and branch it out into its 10 to 12 individual compounds and see which one of those compounds gives the best response. So this is kind of like a preliminary screen, basically how we do our experiments. So with that, that's the technique, and now I'm going to jump right into the data. So the first part, I'm going to talk about floral odor detection, which has an importance in nectar feeding of mosquitoes. So this is a paper that we published in 2019, and it has to do with OR29 from Anopheles gambia. So this receptor is highly expressed in the labella of both male and female mosquitoes. And the labella is at the tip of the proboscis, which is used for feeding both blood and nectar. So linalool is a plant volatile that's closely associated with flowering plants. It's also an enantiomeric compound, meaning that it exists in one of 2 forms in nature. And that is due to a chiral carbon shown here that causes a rotational event to occur. So these are 2 structurally different mirror image molecules. So in this paper, we identified OR29 from Anopheles gambia to be very sensitive to linalool enantiomers. So shown in blue is S plus linalool. That is going to be on the left-hand side of the graph, signifying that this receptor complex responds much more sensitively at lower concentrations to this enantiomer in comparison to R minus linalool. And we get values called EC50 values, which means this much of this compound is required to stimulate the receptor 50% of its maximal response, essentially. So this was my first paper, so that was awesome. Having worked in science for so long with having a lot of failures, this was cool to get this out. And we decided to do some follow-up work. So I'm showing you a phylogenetic tree here of different anopheline mosquitoes. So we have OR29 homologs on the bottom side of this graph. And we have a second receptor on the top, OR53, that is very similar at the amino acid level in comparison to OR29. So they're anywhere from 60 to almost 100% identical at the amino acid level. So we hypothesized that the second receptor is likely doing something similar or detecting a similar type of structurally related compound. So what we went ahead and did was we focused on Anopheles stevensii, OR53 and 29, which is the malarial mosquito primarily in India, and Anopheles gambia, OR29, 53, which is primarily a vector in Africa. So we took a look at these 4 receptors and did the similar types of experiments using the oocyte system. So this data I've already shown you, where Anopheles gambia OR29 responds really well to S plus linalool. So I did the same experiments using Anopheles stevensii OR29 to see, does the same thing happen in this case? And the case was yes, it does. So S plus linalool was on the left-hand side of the graph. It has a lower EC50 value, meaning that this compound, the receptor complex is more sensitive to this enantiomer. So we went ahead and did the same experiments with OR53 from Anopheles gambia. We actually saw a reversal of what we're seeing in this top figure. The black line for R minus is going to be on the left-hand side of the graph, signifying that this receptor complex does a better job of detecting the R- enantiomer. Those lines are very close, so it's hard to say how much of that you would detect or see in a real insect, but when we looked at Anopheles stevensii, we saw the lines spread out much more. So it's basically the exact opposite of what we see for OR29. We're getting really good responses for R- linalool and black in comparison to S+. So these receptors are displaying enantioselectivity. So they're better at detecting one of these two structures in comparison to the other. So which is actually it's fairly unique and has only been described a few times in terms of mosquito sensory biology. So that was really cool. And all of this data here is what Jason was talking about. We have a manuscript that's pretty much ready to submit at this point. So we'll probably be doing that in the next couple of days or so. So this was for floral detection. And the reason why this is interesting and important, just to remind you, is linalool is a plant-derived volatile, and it probably plays some role in the mosquito's ability to detect flowers to take a nectar meal from. So these are two different anopheline mosquitoes that are able to find a flower in nature to take a nectar meal from. So maybe this compound is doing something important, like signifying to these mosquitoes that there's a carbohydrate source available, which that hypothesis obviously requires more research, but that is what's exciting and interesting about this. This information can be used in applications such as attractive toxic sugar rates, which are a mechanism to essentially bait and kill mosquitoes. So inside of this bottle is a sugar source, some sort of attractive odor, and a toxin. So mosquitoes, like shown here on the right-hand side, can feed on this substance, and they will either die due to the toxin or their reproductive capacity will be decreased enough so that you get drops in mosquito population. When you get a drop in mosquito populations, at least in the local environment, you can get decreases in the associated disease transmission that occurs due to these populations. So these experiments allow us to identify novel odor ligands that can either be activators or inhibitors of receptors and mosquito behavior. And this information can be used in combination with one another. So if we're able to mask human scent or push mosquitoes away from a blood meal host, such as a human, and pull them into a trapping device, we're able to decrease the amount of disease that is transmitted by mosquitoes in a local environment. And this idea is called push-pull vector control, and it's been used successfully in different publications. Okay, so I am now showing you a large phylogenetic tree of about 95 ORs from 4 different mosquito species. This is a figure from one of our recently awarded NIH research grants. So on the right-hand side, we have different chemicals that are already known to be excitatory to these specific ORs. So when we look at these amino acid sequence identity between all of these receptors, they cluster with one another so that we hypothesize that these receptors that don't have a compound next to them are going to likely be detecting some sort of floral odor due to the amino acid sequence similarity that we display throughout this tree. So we've begun working on this large research effort, and so far we've been really successful. So the first bit of data I'm going to show you is about this plate of receptors here, shown here. So from 3 different mosquito species that all detect the same compound. So this is a concentration response experiment for Aedes aegypti OR49. So we did the same oocyte experiments, and this receptor responds really highly with low EC50 values to plus camphor. The same thing held true for OR49, which is in that same plate of receptors. And interestingly enough, even a mosquito in a completely different genus has a homologue for this gene, OR38, and it detects the same compound. So there's some evolutionary layeredly conservation that is being displayed to detect this compound camphor. So it's another enantiomeric compound. And it's a floral odor, so it's highly prevalent in the tree, the camphor tree, so that's where it gets its name. And what's pretty cool about this is this compound is actually already known to be a repellent to many different mosquitoes specimens. It's just the molecular reason why that happened wasn't known before. So we hypothesize that the interaction between these receptors and this compound is probably going to be the mechanism for this behavioral output that is already known to the literature. So that was cool to kind of start out and already get some really good data, but we've been fortunate enough and have actually produced a few other deorphanizations. So I'll go ahead and start with this one here, OR13 from Aedes aegypti, which is in this cluster right there, with OR37. So this receptor complex does a really good job of detecting this compound P. cressyl. So it gives low micromolar EC50 values. And we have an additional QLEX homolog that is doing the same molecular detection of this compound. And it even gives nanomolar EC50 values, which was, I wouldn't say surprising, but extremely encouraging to get these results. What's even cooler about these interactions is we have collaborators on this research grant. So it's Omar Akbari from UC San Diego and Jeff Riffle from the University of Washington. They've actually performed some behavioral experiments in living mosquitoes. And when P. cressyl is a component of a floral odor blend, it is highly attractive to different species of Aedes aegypti in particular, and even an Anopheline mosquito, Anopheles stevensi. So that's what is shown here in the green bar. When P. cressyl is a component of this blend, it's highly attractive. So that was really cool to get some behavioral evidence that this is an important interaction. And we've also been even more successful so far. We have OR15 from Aedes aegypti, which is up here at the top, that we have identified to detect 1-hexanol in low micromolar EC50 ranges. We've also identified Aedes aegypti OR28, which is in this clade right there, that detects linalool derivatives, which is really interesting. These compounds have extremely similar structure, but so it makes sense that it might do a good job of detecting both enantiomers of linalool as well as tetrahydrolinalool. So this is cool because this is similar to the data I just showed you on Anopheles, OR29. That kind of does a similar type of thing. And again, our collaborators from UCSD and University of Washington have done some really cool behavioral experiments. So this is a dual choice olfactometer. What you do is you release a bunch of mosquitoes in this chamber here. You have an odor cue on one side and a blink control cue on the other. And you basically assay the number of mosquitoes that go to this arm or this arm, and you could get a preference index of this. So what they did was they have a semi-proprietary blend of odors. But what's important is this star and this star are going to be 1-hexanol and linalool derivatives, respectively. So if we look on the right-hand side of the graph, when both of these compounds are present, you get the best preference index or the most attractive odor cue. And they've also done a number of other experiments where they add and subtract certain odors in order to see what is the best blend. But needless to say, when both of those compounds, as well as 2 mystery compounds, are present, you get the most attractive odor blend. So again, behavioral data to support the molecular interactions that we've identified in this. All right, so that was all about mosquito floral odor detection. So now I want to introduce to you this compound indole. Indole is a nitrogen-containing aromatic compound, and it's known to be important in different flower or plant odors, as well as it can actually be a component of human odor or blood meal host odor. So it might have a number of different interesting interactions in how insects behave. There's also evolutionary conservation of being able to detect this compound. So in mosquitoes on the bottom here, there's already 3 receptors that are known to detect indole, and the second indole-like compound, 3-methyl-indole. And on the top side of this figure, we have a more basal lineage of flies, which can include things like house flies and fruit flies. And they encode receptors that are homologs to these 3 receptors. So we decided to go ahead and go forward and try to deorphin these receptors in the housefly Musca domestica. So we're kind of switching gears. We're going to be talking about houseflies for a second. So I just want to tell you why those are important. So houseflies, as you might know, are pretty nasty organisms. They breed and live in unsanitary conditions. They're mechanical vectors of different diseases, such as bacteria, fungi, and viruses. And there's a big old huge long list if you want to look in the literature, like hundreds of organisms that are thought to be mechanically vectored by houseflies. When I was doing this research, I found a really cool paper that I just want to tell you about real quick, because I'm sure you'll find it interesting. There is a group that did some work on mechanical vectoring of SARS-CoV-2 virions by houseflies, and they've were able to detect virions over the surface of a housefly as well as the intestinal tract of houseflies. So these flies defecate all over the place essentially when they land. So this could be a mechanism for being able to move SARS-CoV-2 virions throughout the environment. Just something I thought was cool that would be worth mentioning. So now I'm going to jump right into the data for our housefly data that we have. So I performed oocyte experiments with one of those indole detecting homologs we thought we identified. So this one's OR30A. It does a really good job of detecting this compound here, which is 3-methyl-indole. And then we took one of those other homologs and did the same experiments. In this case, it's OR49B. And this receptor does a really good job of detecting indole. So that was great. We wanted to take a look at how sensitive are these receptors to different concentrations of these compounds. So we did experiments where we perfused different concentrations of these odors across oocytes expressing our receptors. And in the case of OR30A, we get nanomolar EC50 level, EC50 values for OR30A for 3-methyl-indole, which is this compound that has a methyl group at this carbon position. And then we also perfused indole and 2,3-dimethyl-indole across these oocytes, and we got a more than 10-fold increase in the EC50 value, signifying that the interaction between 3-methyl-indole and this receptor is going to be more ecologically important because it detects it at lower concentrations. So I did the same experiments with OR49B using indole, which gave us low nanomolar concentrations. And when we perfused 3-methyl-indole, we get about a 10-fold increase in EC50 value, just like we saw in that previous graph. It's interesting to think about the binding of a ligand and a receptor. So if you think of it binding into some sort of 3-dimensional pocket, when you add just a methyl group onto this, basically the exact same compound, you get a dramatic reduction in the ability of this receptor to detect or bind this compound. So that's just really interesting to think about 3-dimensionally. And there are some research efforts trying to understand that a bit more in detail. So all of this information we published in Insect Biochemistry and Molecular Biology just this past year. And this is ongoing research in the lab. So another graduate student, John, is going to be taking a look at this in a little bit more detail to see what sort of behavioral relevance these compounds might have, as well as deorphining other housefly odorant receptors that might have an importance in other behavioral traits. So far I've talked mainly about different floral odors or plant-associated odors, and now we're going to kind of switch gears and start talking about host odor detection by mosquitoes. So we published a paper in 2020, I believe, about carboxylic acid responses by a conserved odorant receptor. So what are carboxylic acids? Well, they are compounds that are known to be components of human odor and other animal odors. And they're attractive to different insects, especially mosquitoes. So they could be a cue to these insects that a blood meal host is nearby to take a blood meal. So what we did in this paper is we identified two receptors, OR48 from Aedes aegypti in red and Aedes albopictus in gray, that respond really well to carboxylic acids that are medium to long to kind of long in length. So these are the compounds here. So we have hexanoic acid, which has 6 carbons, 7, 8, 9, 10, all the way to 14 carbons, tetradecanoic acid. And we get this response profile. So as the carbon chain increased from left to right, we started getting higher and higher responses, with the best responses happening for dodecanoic and tridecanoic acid, which are 12 and 13 carbon long in respectively. So we took a look at these receptor sensitivities to these two compounds. So for dodecanoic acid, they both do a really good job of detecting this compound. We get EC50 values in the low micromolar range, which has been a constant theme throughout this presentation, which is good data. It tells us that this is likely something important physiologically. And then we also did the same set of experiments with tridecanoic acid and got similar results. Again, low micromolar levels of EC50 values. So this was, you wouldn't really appreciate this unless you study insect chemosensory biology like we do, but this was actually, to our knowledge, the first time someone's deorphined an odorant receptor or an OR that detects this class of molecule, so a carboxylic acid. They're typically detected by a completely different class of receptors, which I'm going to go ahead and start talking about now. So we have another NIH-funded grant that takes a look at the second class of receptors, which is IRs. So we've identified another clade or group of receptors in different mosquito specimens that we believe likely have an important implication in detecting carboxylic acids, which are, again, a component of human sweat. So we started working on this tree, and we've been lucky enough to get a few deorphins so far. So in this group of receptors here, we've identified IR75E homologs from Aedes albopictus, which is the Asian tiger mosquito, and Aedes aegypti, the yellow fever mosquito, that detect this compound nonanoic acid really well. In red is going to be the Anopheles gambia ortholog IR75K, which is shown here on the graph. And this was actually previously reported to detect this compound by Jason's previous lab with him and his research people that he worked with. So also in this group, we have Aedes aegypti IR75E, which I'm showing you here, that interestingly enough does a really good job of detecting two compounds, octanoic acid and nonanoic acid, which I'm showing you down here, the structures. One has 8 carbons, the other has 9. So it does a really good job, and that was interesting and something we didn't expect to find. So this work is continuing to hopefully deorphin all of the receptors you see there so that we get important information about how mosquitoes might be able to detect blood meal hosts. So I just kind of want to give you a quick summary because I know that was kind of a lot of information I just threw at you. So I'll kind of summarize what I've presented so far. So in terms of nectar feeding or plant odor detection, I talked to you about Anopheles gambia and Stevensi OR29 and 53 that detect linoleuol luol. And then I went on to talk about some culisine orthologs that detect the molecule camphor and its behavioral relevance, as well as OR13 and 38 from these same two mosquito species that detect pea crestle. And then I talked about OR15 and OR28 from Aedes aegypti and how it detects these two behavioral relevant compounds. And then we kind of switched gears a little bit and started talking about the housefly ORs that detect indole-like compounds and how we don't quite know yet what their importance is in terms of behavior, but it might play a role in finding food sources or ovipositing their eggs. And then we started talking about blood meal host seeking by mosquitoes. We talked about that OR pair that detects dodecanoic and tridecanoic carboxylic acids. We moved on to the IR family of chemoreceptors that detect nonanoic acid, or IR75E, and then IR75F that detected octanoic and nonanoic acid very well in our oocyte system. So that's kind of what I've talked to you about so far, which has kind of included all these different molecular interactions that we think might have a really important part to play in the chemical ecology of these mosquitoes and other insects that we talked about. And now I'm going to switch gears and talk about mosquitoes themselves. So I'm going to go into a little bit of how we identify species of mosquitoes and the different ways we can do that. So this paper was published just this last month that identified an anopheline mosquito that actually is comprised of 3 different species of mosquito that are so morphologically identical, so they look the same that you wouldn't be able to tell that they're actually different species of mosquito. So this brought the recognized number of mosquitoes to almost 3,600. And these researchers utilize complex morphological features to identify these 3 different species. So I don't know about you, but I wouldn't be able to tell that these are 3 different species of mosquitoes looking at these small hairs that are extremely difficult to see, or the different ways the wings look under a microscope. Needless to say, this requires a lot of expertise that is really hard to accumulate. It's time consuming and it can be very laborious. You have to look at each individual mosquito one at a time to try to identify. So there's an alternative method, and that is to utilize DNA barcoding. So what DNA barcoding is you take a gene such as cytochrome C oxidase subunit 1, which is a really well-conserved mitochondrial gene that eukaryotes all encode. And because it does such a specific function, all organisms share a very similar DNA sequence of this gene. So what we did was we took the sequence of this gene from 8 different genera of mosquitoes, performed a sequence alignment, and took a look at where are the differences in this gene. So the residues that are highlighted in blue are polymorphisms that occur. If it's gray, it's conserved throughout. So we designed some primers to amplify this region of DNA by PCR. So the idea is you take a mosquito or even just a leg of a mosquito, you throw it inside of a tube with some buffer and you vortex it. And this dissociates the cells enough so that you could amplify mitochondrial DNA using PCR and these degenerate primers. So you could run your amplicons out on a gel, make sure you had amplification occur, and you could clone or directly sequence these amplicons using DNA sequencing such as Sanger sequencing. The sequencing results you get back, you simply just throw into NCBI BLAST and you identify what is the best match that you get. So there are some limitations with this. Not every mosquito is going to have a reference sequence in the database itself. So this method is only as good as what you're blasting against, right? If your mosquito specimen is brand new and it's never been found before or cataloged, you're not going to get a very good match. But that's why publications usually use a certain level of nucleic acid identity as a cutoff. So if it's 98% similar, we can call it this genus. If it's 99% similar, we could call it this subspecies, etc. It's kind of how this works throughout. Not only insects, but this method is used for identifying all types of organisms, particularly invertebrates. So the idea behind this is, let's say we have 2 populations of a mosquito that kind of are isolated from one another. And throughout time, enough time and evolutionary change undergoes between these 2 populations that they are no longer able to mate with one another. And that is the definition of the species, right? Being reproductively isolated. But they still look exactly the same.
So not enough evolution has occurred. So that is where this DNA barcoding comes into, becomes handy, is because maybe we can't tell the difference of these mosquitoes morphologically, but maybe we'll get a much better idea if we take a look at their genetic material to see if they're different species. So that's the idea behind this. And we worked with some collaborators from New Mexico State University primarily, that went to Malaysia and Borneo, and they collected mosquitoes in different regions of this island. So that's what I'm showing you here. All these different dots are their different collection sites, and based on color is kind of what kind of land this trapping occurred in. Was it urban? Was it a forest? Was it kind of a rural area? What was the ecosystem and the habitat like? And what we were able to do using this method of identifying mosquitoes, both morphologically, but also getting a second confirmation molecularly of their species, is, let's say we're looking at a village and there's 3 different species of mosquito in this village because we identified them based on their sequence and their morphology. We can do what our collaborators did, is take a look at what are these mosquitoes feeding on. So when they collect them, they have blood inside their abdomen, and we could use the same DNA barcoding approach to identify where that blood came from. What animal did it come from? Did it come from this species of boar? Did it come from a human or this species of boar? And then we could use that information in combination with one another to try to determine vector host blood feeding patterns that can have an importance in the transmission of different pathogens that are vectored by these mosquitoes. Maybe this orange mosquito shown in the middle that feeds on both humans and animals doesn't transmit our pathogen of interest, but this mosquito in green does. The orange mosquito does not have the capacity to transmit this disease, but maybe this more important one does, that actually feeds on humans and animals. So that's important information to know. And if you also take into account the different sites that were sampled from, you could also see how does vector-host relationships change spatially throughout the environment. So if we look in a rural buildup area, do we see the same types of mosquitoes feeding on the same types of things? So that was a large portion of what this paper was, but we definitely played a key role in being able to confirm the identity of the mosquitoes that we collect. So this technique does have some limitations, like I mentioned before, but it is, when used in combination with other techniques, and even using multiple DNA barcoding genes, you can get a much better confirmation of species identity. So I think with that, that kind of wraps up everything I want to show you today. Of course, want to thank the lab that have always been extremely supportive. Basically, everyone's here right now. Jason, I mean, everyone that knows him in this room. I really don't know what to say. I could not have gotten a better PhD advisor. Both personally and professionally, you allow us to perform the research that we want to do, and kind of allow us the intellectual freedom to, do what we want to do on our own until we need your help. And you're okay with maybe it taking a week or 2 longer, but we did it on our own. And just the opposite is true. If someone likes working more hands-on and like close with you, you're completely amenable to that. And I mean, there's a million other things I could say, but I will not do that so that I don't get emotional. But thank you. It has honestly it's been amazing. So hopefully I get to embarrass you a little bit, because you did it to me. I'm not done yet, but I want to thank my committee also. Dr. Carter, Dr. Saluki, Sim, and Tavi, thank you all for being or agreeing to be on my committee. I don't know if you wanted to or not, but I appreciate it, and I've gotten really good feedback from everyone throughout this process. I'd also like to thank our collaborators. That's another great thing about the lab, is we work with so many different people throughout the world, not even just the country or the state. So Jonathan Bobot is from the Hebrew University of Jerusalem. Omar Akbari is at UC San Diego, where I'll be transitioning to. Jeff Riffle is at University of Washington. Matt DeGennaro is at Florida International University, and Dr. Catherine Hanley is at New Mexico State University. And those are just the collaborators that I worked with, not to mention the other PhD students in the lab. So that's another great thing, is when you're able to meet and work with so many different people. Of course, I wouldn't have been anywhere without my family. Everyone's been incredibly supportive throughout all of this. That is my brother, Justin, on the left, his wife, April, my dad, Robert, my mom, Christy, my sister, Janelle, her new fiance, Calais, and their beautiful baby girl, Vivian, who's my niece. And my friends from San Diego, these two guys have been incredibly supportive, especially these last few months. So that's Chad Gong. He might be watching right now. I've known this guy. I basically grew up with him. And Alex Padua, I met 12 years ago, like my first day of college. So still friends with both of them. This is us at the shooting range here in town when he came for a few days. Now I will try not to get too sentimental because I don't want to embarrass myself. But this is my master's thesis advisor, Roland. He was probably the most passionate person I've ever met in my entire life. He gave me an opportunity to work in his lab as an undergraduate. I had no idea what I was doing or what I wanted to do in school. And he gave me a shot and kind of taught me how to not be scared to ask questions and to just be yourself. And unfortunately, Roland passed away in April of 2020. And science lost a really great person. And I just wanted to kind of make sure everyone knows that, how special he was. And yeah, I wouldn't be here if it wasn't for him. So I had to do that. Yeah, I'm almost crying. It's okay. He was a great person. I mean, this picture on the left, that is him attending his graduate students' graduation with terminal lung cancer. So that's just the kind of person he was. He cared so much for his students and would do anything for them. So I just had to say thank you. Also, on a happier, lighter note, I've had a really great privilege to be here. I mean, I was not a very good candidate as a PhD candidate on paper. I think Jason and maybe some others during the interview weekend, you know, kind of decided to give me a chance. And I'm so happy that I got to do this. And with that, I would be happy to answer any questions.
Yeah, so I think we have plenty of time for some questions. We can take them from the room, of course, and we can also take them from the public who are on Zoom, who aren't in the room. Maybe, Joe, we might need help with the questions on Zoom because I don't think I can hear them here. Yeah, so if anyone has a question, if you want to raise your hand and Zoom.